Regardless sensor has kinase regulates ARR4 activity, the studies by Sphere and colleagues suggest a novel type of crosstalk between an ancestral two-component system and the phytochrome B-dependent light signaling pathway, figure 4. This crosstalk enables the integration of signal information provided by hormone, cytokinin, and red light phytochrome B signal transduction cascades. It is now of major interest to identify the effector proteins of the remaining type of response regulators and to clarify the underlying crosstalk mechanisms. A complex plant two-component signaling network emerges. Conclusions and perspectives. As described above, there is now the first experimental evidence that hybrid kinases like CRE1, AHK2, and AHK3 signal through multi-step phosphorylase containing AHPs and ARRs. In addition, a wealth of these two hybrid and in vitro protein-protein interaction studies indicate the possibility of a complex two-component signaling network in cells of higher plants. A detail of such a potential network is shown in Figure 5. Specificity to some of these interactions can be recognized. For example, ETR1 and CRE1 interact with both AHP1 and AHP2, whereas CKI1 interacts with only AHP2, Figure 5. Specificity is also apparent for the interaction of ARRs with AHPs. ARR2 interacts with AHP1 and AHP2, whereas the interaction of ARR4 is restricted to AHP1, Figure 5. As indicated in previous sections, two-component signaling elements from Arabidopsis can participate in phosphorylase from biologically irrelevant fungal systems. However, even given the preliminary nature of this emerging picture, crosstalk between different two-component signaling pathways would have major functional implications. Two-component systems may form a complex signaling network capable of sensing, computing, and integrating a broad range of signals. The lifestyle of plants makes it necessary that every cell monitors and integrates a multitude of inputs to reach a finely balanced decision on how to react at the cellular and the whole plant level. This potential for crosstalk potentially represents an additional reason as to why multi-step two-component systems rather than simple two-component systems were utilized in plants. We have already received our first glance at this complexity from studies revealing crosstalk between the ARR4 comprising two-component system with phytochrome B-dependent signal transduction. Overlapping sets of synergistic and antagonistic physiological responses are also described for many other stimuli such as ethylene and cytokinin. Complexly interconnected two-component systems could participate in the generation of these overlapping physiological responses. Figure 5. Detail from the emerging network of two-component signaling systems in Arabidopsis. Lines indicate direct protein-protein interactions detected in the yeast 2 hybrid system and in vitro. Proteins that are not connected do not display physical interaction. ETR1, CRE1, and CKI hybrid kinase suggested to be involved cytokinin signaling. AHP1 and AHP2, HPT domain proteins. ARR2 and ARR4, response regulators. The conserved amino acid residues H, His, T, Asp of the different two-component modules are indicated. C. Cell-cell interactions. The extracellular matrix. All cells are not surrounded by cell walls. Many of the cells and tissues of multicellular organisms are embedded in an extracellular matrix consisting of secreted proteins and polysaccharides. The extracellular matrix fills the spaces between cells and binds cells and tissues together. One type of extracellular matrix is exemplified by the thin, sheet-like basal laminae, or basement membranes, upon which layers of epithelial cells rest. Figure. In addition to supporting sheets of epithelial cells, basal laminae surround muscle cells, adipose cells, and peripheral nerves. Extracellular matrix, however, is most abundant in connective tissues. For example, the loose connective tissue beneath epithelial cell layers consists predominantly of an extracellular matrix in which fibroblasts are distributed. Other types of connective tissue, such as bone, tendon, and cartilage, similarly consist largely of extracellular matrix, which is principally responsible for their structure and function. Extracellular matrices are composed of tough fibrous proteins embedded in a gel-like polysaccharide ground substance. In addition to fibrous structural proteins and polysaccharides, the extracellular matrix contains adhesion proteins that link components of the matrix both to one another and to attached cells. The major structural protein of the extracellular matrix is collagen, which is the single most abundant protein in animal tissues. The collagens are a large family of proteins, containing at least 19 different members. They are characterized by the formation of triplet alpha helices in which 3 polypeptide chains are wound tightly around one another in a rope-like structure. Collagen molecules assemble in a regular staggered array to form fibrils. The molecules overlap by 1/4 of their length, and there is a short gap between the N-terminus of one molecule and the C-terminus of the next. The assembly is strengthened by covalent crosslinks between side chains of lysine or hydroxylase residues, primarily at the ends of the molecules. Connective tissues also contain elastic fibers, which are particularly abundant in organs that regularly stretch and then return to their original shape, e.g. lungs. Elastic fibers are composed principally of a protein called elastin, which is cross-linked into a network by covalent bonds formed between the side chains of lysine residues, similar to those found in collagen. The fibrous structural proteins of the extracellular matrix are embedded in gels formed from polysaccharides called glycosaminoglycans, or GAGs, which consist of repeating units. The fibrous structural proteins of the extracellular matrix are embedded in gels formed from polysaccharides called glycosaminoglycans, or GAGs, which consist of repeating units of disaccharides. One sugar of the disaccharide is either N-acetylglucosamine or N-acetylgalactosamine, and the second is usually acidic, either glucuronic acid or iduronic acid. Hyaluronan is the only GAG that occurs as a single long polysaccharide chain. All of the other GAGs are linked to proteins to form proteoglycans, which can consist of up to 95% polysaccharide by weight. In addition to being components of the extracellular matrix, some proteoglycans are cell surface proteins that function in cell adhesion. E proteins, the third class of extracellular matrix constituents, are responsible for linking the components of the matrix both to one another and to the surfaces of cells. The prototype of these molecules is fibronectin, which is the principal adhesion protein of connective tissues. Fibronectin is a dimeric glycoprotein consisting of 2 polypeptide chains, each containing nearly 2,500 amino acids. In the extracellular matrix, fibronectin is further cross-linked into fibrils by disulfide bonds. Fibronectin has binding sites for both collagen and GAGs, so it cross-links these matrix components. A distinct site on the fibronectin molecule is recognized by cell surface receptors and is thus responsible for the attachment of cells to the extracellular matrix. Basal laminae contain a distinct adhesion protein called laminin. Laminins can self-assemble into mesh-like polymers. Such laminin networks are the major structural components of the basal laminae synthesized in very early embryos, which do not contain collagen. The major cell surface receptors responsible for the attachment of cells to the extracellular matrix are the integrins. The integrins bind to short amino acid sequences present in multiple components of the extracellular matrix, including collagen, fibronectin, and laminin. Direct cell-cell interactions. Direct interactions between cells, as well as between cells in the extracellular matrix, are critical to the development and function of multicellular organisms. Some cell interactions are transient, such as the interactions between cells of the immune system and the interactions that direct white blood cells to sites of tissue inflammation. In other cases, stable cell-cell junctions play a key role in the organization of cells and tissues. For example, several different types of stable cell-cell junctions are critical to the maintenance and function of epithelial cell sheets. Cells also associate with their neighbors not only by interactions between their cell walls, but also by specialized junctions between their plasma membranes. Cell adhesion proteins. Cell-cell adhesion is a selective process, such that cells adhere only to other cells. Cell adhesion proteins. Cell-cell adhesion is a selective process, such that cells adhere only to other cells of specific types. Such cell adhesion is mediated by transmembrane proteins called cell adhesion molecules, which can be divided into four major groups: the selectins, the integrins, the immunoglobulin Ig superfamily, it's named because they contain structural domains similar to immunoglobulins, and the cadherins. The selectins mediate transient interactions between leukocytes and endothelial cells or blood platelets. The selectins recognize cell surface carbohydrates. One of their critical roles is to initiate the interactions between leukocytes and endothelial cells during the migration of leukocytes from a circulation to sites of tissue inflammation. The selectins mediate the initial adhesion of leukocytes to endothelial cells. This is followed by the formation of more stable adhesions, in which integrins on the surface of leukocytes bind to intercellular adhesion molecules, ICAMs, which are members of the Ig superfamily expressed on the surface of endothelial cells. The attached leukocytes are then able to penetrate the walls of capillaries and enter the underlying tissue by migrating between endothelial cells. The fourth group of cell adhesion molecules, the cadherins, also display homophilic binding specificities. They are not only involved in selective adhesion between embryonic cells but are also primarily responsible for the formation of stable junctions between cells and tissues. For example, E-cadherin is expressed on epithelial cells, so homophilic interactions between E-cadherins lead to the selective adhesion of epithelial cells to one another. It is noteworthy that loss of E-cadherin can lead to the development of cancers arising from epithelial cells, illustrating the importance of cell-cell interactions in controlling cell behavior. Members of the cadherin family, such as N-cadherin, neurocadherin, and P-cadherin, placental cadherin, mediate selective adhesion of other cell types. Stable cell-cell junctions mediated by the cadherins. Homophilic interactions between cadherins mediate two types of stable cell-cell adhesions. In adherens junctions, the cadherins are linked to bundles of actin filaments via the tenins. In desmosomes, desmoplakin links members of the cadherin superfamily, desmoglanes and desmoglins, to intermediate filaments. Tight junctions, which are usually associated with adherens junctions and desmosomes in a junctional complex, are critically important to the function of epithelial cell sheets as barriers between fluid compartments. Tight junctions play two roles. First, tight junctions form seals that prevent the free passage of molecules, including ions, between the cells of epithelial sheets. Second, tight junctions separate the apical and basolateral domains of the plasma membrane by preventing the free diffusion of lipids and membrane proteins between them. Specialized transport systems in the apical and basolateral domains are able to control the traffic of molecules between distinct extracellular compartments. Tight junctions are the closest known contacts between adjacent cells. They were originally described as sites of apparent fusion between the outer leaflets of the plasma membranes, although it is now clear that the membranes do not fuse. Instead, tight junctions appear to be formed by a network of protein strands that continues around the entire circumference of the cell. Each strand in these networks is thought to be composed of transmembrane proteins, claudins and occludin, that bind to similar proteins on adjacent cells, thereby sealing the space between their plasma membranes. Gap junctions, which are found in most animal tissues, serve as direct connections between the cytoplasms of adjacent cells. They provide open channels through the plasma membrane, allowing ions and small molecules less than approximately 1,000 daltons to diffuse freely between neighboring cells, but preventing the passage of proteins and nucleic acids. Consequently, gap junctions couple both the metabolic activities and the electric responses of the cells they connect. Most cells in animal tissues, including epithelial cells, endothelial cells, and the cells of cardiac and smooth muscle, communicate by gap junctions. In electrically excitable cells, such as heart muscle cells, the direct passage of ions through gap junctions couples and synchronizes the contractions of neighboring cells. Gap junctions allow the passage of some intracellular signaling molecules, such as CAMP and Ca2+, between adjacent cells, potentially coordinating the responses of cells and tissues. Gap junctions are constructed of transmembrane proteins called connexins. 6 connexins assemble to form a cylinder with an open aqueous pore in its center. Such an assembly of connexins in the plasma membrane of one cell then aligns with the connexins of an adjacent cell, forming an open channel between the two cytoplasms. The plasma membranes of the two cells are separated by a gap corresponding to the space occupied by the connexin extracellular domains, hence the term gap junction, which was coined by electron microscopists. D1. Uncontrolled cell division leads to cancer. In the normal circumstances, the proliferation of body cells is under strict control. The cells differentiate, divide and die in a sequential manner in a healthy organism. Cancer is characterized by loss of control of cellular growth and development, leading to excessive proliferation and spread of cells. Cancer is derived from a Latin word meaning crab. It is that the word cancer originated from the character of cancerous cells, which can migrate and adhere and cause pain, like a crab, to any part of the body. Neoplasia literally means new growth. Uncontrolled growth of cells results in tumors, a word originally used to represent swelling. Oncology Greek, onkos tumor, deals with the study of tumors. The tumors are of two types. A. Benign tumors. They usually grow by expansion and remain encapsulated in a layer of connective tissue. Normally benign tumors are not life-threatening, e.g. moles, warts. These types of benign tumors are not considered as cancers. B. Malignant tumors are cancers. They are characterized by uncontrolled proliferation and spread of cells to various parts of the body, a process referred to as metastasis. Malignant tumors are invariably life-threatening, e.g. lung cancer, leukemia. About 100 different types of human cancers have been recognized. Cancers arising from epithelial cells are referred to as carcinomas, while that from connective tissues are known as sarcomas. Methods for the early detection and treatment of cancers have been developed. However, little is known about the biochemical basis of cancer. Metastasis. Metastasis refers to the spread of cancer cells from the primary site of origin to other tissues of the body, where they get deposited and grow as secondary tumors. Metastasis is the major cause of cancer-related morbidity and mortality. The biochemical basis of metastasis is not clearly known. It is biological changes in tumor cells, loss of contact inhibition, loss of anchorage dependence and alterations in the structure of certain macromolecules are among the important factors responsible for metastasis. Metastasis is genetically controlled. Cancer cells cannot regulate their growth and division. As a result, they develop into malignant tumors that may potentially invade neighboring tissue. Sometimes, cancer cells detach from the primary tumor and settle elsewhere in the body, where they grow and divide, producing secondary tumors. This process, called metastasis, is often the cause of death in cancer patients. Metastatic cancer cell spreads from the primary tumor by entering the blood or lymphatic circulatory system. These cells are carried into circulation until they lodge in a capillary bed. Normally, more than 99% of the cells die. The surviving cells invade tissue adjacent to the capillary bed and begin dividing to form a secondary tumor. To reach a new site, the tumor cells pass through the layer of epithelial cells lining the interior wall of the capillary or lymph vessel and penetrate the surrounding extracellular matrices. Extracellular matrix is a meshwork of proteins and carbohydrate molecules separating tissues. It is a scaffold for tissue growth and inhibits the migration of cells. To establish a secondary tumor, metastatic cells secrete enzymes that digest this meshwork, creating holes through which they can migrate. The ability to invade extracellular matrix is also a property of some normal cell types. Implantation of the embryo in the uterine wall during pregnancy, for example, requires cell migration across the extracellular matrix. White blood cells reach the site of infection by penetrating capillary walls. The mechanism of invasion is probably the same in these cells as in cancer cells. The difference is that in normal cells, the invasive ability is controlled and tightly regulated, while in tumor cells, regulation has been lost. Metastasis is regulated by genes that encode or regulate matrix-cutting enzymes. Protein-cutting enzymes called metalloproteinases are necessary for cell invasion. Tumor cells with high metastatic capacity produce more metalloproteinases than do tumor cell lines with low invasive properties. Activated metalloproteinases are inhibited by the presence of a tissue inhibitor of metalloproteinase TIM. Normal cells produce TIMP and thereby suppress inappropriate activity of metalloproteinase. In tumor cells, TIMP inhibits metalloproteinase only when the number of TIMP molecules is greater than the number of metalloproteinase molecules. On the basis of its action and regulation, TIMP can be considered as a protein that suppresses metastasis. Incidence: cancer is the second largest killer disease, the first being coronary heart disease, in the developed countries. It is estimated that cancer accounts for more than 20% of the deaths in the United States. Based on the current rate of incidence, it is believed that 1 in every 3 persons will develop cancer at some time during his life. Although humans of all age develop cancer, the incidence increases with advancement of age. More than 70% of the new cancer cases occur in persons over 60 years. Surprisingly, cancer is a leading cause of death in children in the age group 3 to 13 years, half of them die due to leukemia. Etiology: in general, cancers are multifactorial in origin. The causative agents include physical, chemical, genetic and environmental factors. Survey in USA has shown that about 90% of all cancer deaths are due to avoidable factors such as tobacco, pollution, occupation, alcohol and diet. Most of the cancers are caused by chemical carcinogens, radiation energy and viruses. These agents may damage DNA or interfere with its replication or repair. 1. Chemical carcinogens. It is estimated that almost 80% of the human cancers are caused by chemical carcinogens in nature. The chemicals may be organic, e.g. dimethylbenzanthracene, benzo-pyrene, dimethylnitrosamine, or inorganic, arsenic, cadmium, in nature. Entry of the chemicals into the body may occur by one of the following mechanisms. 1. Occupation, e.g. asbestos, benzene. 2. Diet, e.g. aflatoxin B produced by fungus, Aspergillus flavus, contamination of foodstuffs, particularly peanuts. 3. Drugs. Certain therapeutic drugs can be carcinogenic, e.g. diethylstilbestrol. 4. Lifestyle, e.g. cigarette smoking, includes benzo-pyrene and polonium. A. Mechanism of action. Although a few of the chemicals are directly carcinogenic, majority of them require prior metabolism to become carcinogenic. The enzymes such as cytochrome P450 responsible for the metabolism of xenobiotics are involved in dealing with the chemical carcinogens. In general, the chemically non-reactive procarcinogen is converted to an ultimate carcinogen by a series of reactions. The carcinogens can covalently bind to purines, pyrimidines and phosphodiester bonds of DNA, often causing unrepairable damage. The carcinogens frequently cause mutations, a change in the nucleotide sequence of DNA, which may finally lead to the development of cancer, hence they are regarded as mutagens. B. Ames assay. This is a laboratory test to check the carcinogenicity of chemicals. Ames assay employs the use of a special mutant strain of bacterium, namely Salmonella typhimurium his. This organism cannot synthesize histidine, hence the same should be supplied in the medium for its growth. Addition of chemical carcinogens causes mutations, reverse mutation, restoring the ability of the bacteria to synthesize histidine, his plus. By detecting the strain of Salmonella, his plus, in the colonies of agar plates, the chemical mutagens can be identified. The Ames assay can detect about 90% of the chemical carcinogen. This test is regarded as a preliminary screening procedure. Animal experiments are conducted for the final assessment of carcinogenicity. C. Promoters of carcinogenesis. Some of the chemicals on their own are not carcinogenic. Certain substances known as promoting agents make them carcinogenic. The application of benzo-pyrene to the skin, as such, does not cause tumor development. However, if this is followed by the application of croton oil, tumors will develop. In this case, benzo-pyrene is the initiating agent while croton oil acts as a promoting agent or promoter. Several compounds that act as promoting agents in various organs of the body have been identified. These include saccharin and phenobarbital. 2. Radiation energy, ultraviolet rays, X-rays and Y. Radiation energy, ultraviolet rays, X-rays and Y-rays have been proved to be mutagenic in nature, causing cancers. These rays damage DNA, which is the basic mechanism to explain the carcinogenicity of radiation energy. For instance, exposure to UV rays results in the formation of pyrimidine dimers in DNA while X-rays cause the production of free radicals. These types of molecular damages are responsible for the carcinogenic effects of radiations. 3. Carcinogenic viruses, the involvement of 3. Carcinogenic viruses, the involvement of viruses in the etiology of cancer was first reported. 3. Carcinogenic viruses, the involvement of viruses in the etiology of cancer was first reported by Rouse in 1911. He demonstrated that the cell-free filtrates from certain chicken sarcomas, tumors of connective tissues, promote new sarcomas in chickens. Unfortunately, this epoch-making discovery of Rouse was ignored for several years. This is evident from the fact that Rouse was awarded the Nobel Prize in 1966 at the age of 85 for his discovery in 1911. The presence of viral particles in the enzyme reverse transcriptase, besides the occurrence of base sequence in the DNA of malignant cells, complementary to tumor viruses, indicate the involvement of viruses in cancer. Viruses involved in the development of cancer, commonly known as oncogenic viruses, may contain either DNA or RNA. A selected list of tumor viruses is as follows. DNA, the ultimate in carcinogenesis. DNA is the ultimate critical macromolecule in carcinogenesis. Several evidences support this fact. 1. Cancers are transmitted from mother to daughter cells. In other words, cancer cells beget cancer cells. 2. Chromosomal abnormalities are observed in many tumor cells. 3. Damage to DNA caused by mutations often results in carcinogenesis. 4. Laboratory experiments have proved that purified oncogenes can transform normal cells into cancer cells. D3. Molecular basis of cancer. It is clear that the two main properties of cancer, uncontrolled cell division and metastasis, result from mutations. As mentioned earlier, these mutations can involve large-scale genomic instability, chromosome loss, chromosome rearrangement, or the insertion of foreign, often viral, DNA sequences into loci. Small-scale mutations, including nucleotide substitutions or deletions, or more subtle modifications that alter only the amount of a gene product or the time over which it is active may also be involved. In general, control of cell division is regulated in two ways, by genes that normally function to suppress cell division and by genes that normally function to promote cell division. The first group of regulatory genes is called tumor suppressor genes. When expressed, these genes halt passage through the cell cycle and prevent mitosis. For cell division to occur, these genes or their gene products, or both, must be inactive or absent. Suppressor genes become permanently inactivated or deleted through mutation, control over cell division is lost, and the mutant cells begin to proliferate in an uncontrolled fashion. Genes that normally promote cell division are called proteincogenes. These genes can be on or off, and when they are on, they promote cell division. To stop mitosis, these genes or their gene products, or both, must be inactivated. If these genes become permanently switched on, then uncontrolled cell division occurs, leading to tumor formation. Mutant forms of proteincogenes are called oncogenes. In the next sections, we examine how mutations in tumor suppressor genes can lead to a loss of cell cycle control and lead to cancer formation. Then, we consider the role of proteincogenes and oncogenes. In recent years, third categories of genes that control the cell death or apoptosis are also believed to be involved in carcinogenesis. 31.3.1. Tumor suppressor genes normally suppress cell division. Studies have identified a number of genes that, when mutated, confer a predisposition to specific cancers. Table 2. One example of an inherited predisposition to cancer is retinoblastoma, an inherited cancer of the retinal cells of the eye. Retinoblastoma. Retinoblastoma RB occurs with a frequency that ranges from 1 in 14,000 to 1 in 20,000, and most often appears between the ages of 1 and 3 years. Two forms of retinoblastoma are known. Familial form, about 40% of all cases, individuals who carry one mutant RB allele are far more susceptible to developing retinoblastoma than those with two normal wild-type RB alleles. Predisposition to this form of cancer is inherited as an autosomal dominant trait, although the mutation itself is actually recessive, as we will explain later. About 90% of those who inherit the mutant RB allele will. Table 1. Selected tumor viruses class members. DNA viruses. Adenovirus 12 and 18. Herpes virus, Epstein-Barr virus, herpes simplex virus, papavirus, papillomavirus, polyomavirus. RNA viruses. Retrovirus type B, mammary tumor virus of mouse, retrovirus type C, leukemia, sarcoma. Develop retinal tumors, usually in both eyes. In addition, these individuals are predisposed to other forms of cancer, including osteosarcoma, a bone cancer, even if they do not develop retinoblastoma. Second form of retinoblastoma, the remaining 60%, is not familial, and tumors develop spontaneously. This sporadic form is characterized by the appearance of tumors only in one eye, and onset occurs at a much later age than in the familial form. Both types of retinoblastoma. Alfred Knudsen and his colleagues developed a model that requires the presence of two mutated copies of the RB gene in the same retinal cell for tumor formation. In other words, tumor By studying both types of retinoblastoma, Alfred Knudsen and his colleagues developed a model that requires the presence of two mutated copies of the RB gene in the same retinal cell for tumor formation. In other words, by studying both retinoblastoma, Alfred Knudsen and his colleagues developed a model that requires the presence of two mutated copies of the RB gene in the same retinal cell for tumor formation. In other words, tumor development is a recessive trait. In the familial form, one mutant RB allele is inherited and is carried by all cells of the body, including cells of the retina, Fig 4a. If a second RB allele becomes mutated in a retinal cell, retinoblastoma will develop. Therefore, individuals carrying an inherited mutation of the RB gene are predisposed to develop retinoblastoma, as only one additional mutational event is required to cause tumor formation. This does not happen in all cases. About 10% of those inheriting a mutant RB allele do not develop cancer. Presumably, the wild-type allele of the RB gene does not mutate in any retinal cells. In the non-familial sporadic cases, Fig 4b, independent mutations in both normal RB alleles must occur in the same retinal cell for a tumor to develop. As might be expected, these events are far less frequent and occur at a much later age. As predicted by Knudsen's model, such sporadic forms of retinoblastoma are more likely to occur in a single eye. The retinoblastoma RB gene. The retinoblastoma RB gene, located on chromosome 13, encodes a protein designated as PRB. This protein is found in the nuclei of all cell and tissue types examined so far and also is found in both resting GO cells and actively dividing cells. Moreover, PRB is present at all stages of the cell cycle and is part of a regulatory pathway that is important in cell cycle progression. It acts as a molecular switch that controls the passage of cells from G1 into S phase. Because the null gene is expressed ubiquitously in the body, cells can progress through the GS transition only when PRB is inactivated by phosphorylation. PRB is regulated by the action of CDKs. When CDK4 binds to cyclin D, the kinase phosphorylates PRB. PRB is phosphorylated in the S and the G2M stages of the cell cycle, but is not phosphorylated in GO and G1. When PRB is unphosphorylated, its active state, it binds to members of the E2F family of transcription factors. Transcription factors are proteins that bind to the promoter region of genes and regulate transcription. E2F transcription factors control the expression of some 20 to 30 genes required to move the cell from G1 to S in its unphosphorylated form. PRB binds to E2F, blocking transcription, and keeping the cell in G1. Fig 5. The phosphorylation of PRB by CDK4 takes place in late G1. When PRB added, PRB releases E2F, allowing transcription of genes that move the cell from G1 into S. Direct evidence for the role of PRB in regulating the cell cycle comes from several lines of experimental work. Osteosarcoma, bone cancer, cells are homozygous for mutant RB alleles and do not produce PRB. When osteosarcoma cells are injected into a cancer-prone strain of mice, tumors are formed. If a normal RB gene is transferred to the cancer cells, PRE is produced and no tumors are formed when these genetically modified cells are injected into mice. In a separate two-step experiment, a normal RB gene was transferred into osteosarcoma cells. These cells produced PRB and stopped cell division. When D or E cyclins were added to these PRB blocked cells, cell division resumed. Addition of cyclins resulted in phosphorylation of PRB, presumably by activating CDK4 or another kinase. The active PRB stops cell division, that PRB is a target of AG1 CDK, cyclin complex, and that inactivating PRB allows passage through the cell cycle and subsequent division, confirming the role of PRB in G1 control. In normal retinal cells, PRB is active and prevents passage into S by interacting with the transcription factor E2F. In retinoblastoma cells, both copies of the RB gene are mutated, and no functional PRB is present. As a result, E2F activates the genes required for passage through the GS checkpoint into the S phase. This important checkpoint is continually overridden in these cells, resulting in uncontrolled growth and tumor formation. The Wilms tumor gene, Wilms tumor WT, is a kidney cancer found primarily in children. It has a frequency of about 1 in 10,000 births, and, like retinoblastoma, it is found in two forms, a non-inherited sporadic form and a familial form conferring a predisposition to WT. The familial predisposition is inherited as an autosomal dominant trait. According to Knudson's model, familial cases inherit one mutant allele through the germline and develop a mutation in the remaining normal allele in a somatic cell. Such cases, on the other hand, require two independent mutations of the WT gene within the same cell, and tumors more often involve only one kidney. The WT gene is on the short arm of chromosome 11 and encodes a transcription factor. This protein has 4 contiguous zinc finger domains, motifs characteristic of DNA binding proteins that regulate transcription. T has a restricted pattern of expression. It is normally active only in cells of the fetal kidney and only during the brief time when the nephron, the basic filtration unit of the kidney, is being formed. The structure of the WT protein, its pattern and time of expression, and the resultant mutant phenotype have led to the hypothesis that the WT gene encodes a nuclear protein which turns off genes that maintain cell division. Alternatively, the gene product may switch on genes that begin differentiation of the fetal cells into components of the kidney. Cells, the mutant gene is switched on at the appropriate time in the appropriate cells, but the altered gene product is unable to regulate its target genes, resulting in continued cell division, aberrant differentiation, and tumor formation. Although both RB and WT genes encode nuclear proteins that suppress tumor formation, there are some differences in their properties and modes of action. PRB does not bind to DNA, instead, it interacts with E2F, as described in the previous section. WT has all the characteristics of a DNA-binding transcription factor. PRB is expressed in all dividing cells, whereas the WT protein is found only in a restricted period of kidney development. PRB is a general regulator of cell division. The WT protein is a cellular tissue-specific regulator of gene activity during fetal or neonatal development. P53, guardian of the genome. Another tumor suppressor gene P53 has been implicated in human cancer. P53 encodes a nuclear transcription factor. P53 mutations are found in a wide range of cancers, including breast, lung, bladder, and colon cancers. It is estimated 50 to 60% of all cancers are associated with mutations in the P53 gene, suggesting that it controls one or more key events in the proliferation of all cells and is not involved in a cellular tissue-specific form of regulation. Inherent mutations of P53 are associated with the Li-Fraumeni syndrome, an autosomal dominant condition with a predisposition to develop cancers in several tissues at a high frequency. Normally the P53 protein is present in cells at a low level, in a rapidly degraded, and active form. Several types of signals can shut down P53 degradation, causing a rapid increase in P53 protein levels. Chemical damage to DNA, double-stranded breaks in DNA induced by ionizing radiation, or the presence of DNA repair intermediates generated by exposure to ultraviolet light all cause a shutdown of P53 turnover. Active P53 protein can lead to several responses, including 1. DNA repair, 2. Cell cycle arrest, and 3. Apoptosis, a genetically programmed pathway of cell death. These deaths are accomplished by target genes activated by P53 in its role as a transcription factor. The expression of more than 20 target genes are controlled by the action of P53. Cell cycle arrest by P53 can occur at several points in the cycle, including G1. To arrest the cycle at this phase, P53 activates transcription of a CDK inhibitor called P21. The P protein targets a number of CDK cyclin complexes, including the CDK4 cyclin D complex. 21 protein targets a number of CDK cyclin complexes, including the CDK4 cyclin D complex, which was discussed earlier. Inhibiting the CDK4 cyclin D complex keeps PRB in its active configuration, repressing the transcription of genes needed to move the cell from G1 into S phase. Cell functional P53 protein cannot arrest in G1 following irradiation and move immediately from G1 into S. These cells do not repair the DNA damage caused by the radiation and, as a result, have a high rate mutation. Thus, the P53 gene is often referred to as a guardian of the genome. As just mentioned, more than 50% of all human cancers carry a mutation in the P53 gene. The P53 protein is a tetramer, so mutation of one P53 allele usually abolishes all P53 activity, because almost all tetramers will contain at least one defective subunit. As a result, P53 mutations act as dominant negative mutations, and individuals heterozygous for a P53 mutation will develop cancer with a frequency of 90 to 95%. Central role of the p53 gene in cell cycle control emphasizes the relationships between cancer and the cell cycle and between genes that regulate cell growth in cancer. Cancer genes. Mutations in BRCAL, a gene on the long arm of chromosome 17, are associated with a predisposition to breast cancer. This predisposition is inherited as an autosomal dominant trait. About 85% of women carrying one mutant BRCA1 allele will develop a mutation in the second allele and get breast cancer. These women also have an increased risk of ovarian cancer. Second breast cancer gene, BRCA2, on the long arm of chromosome 13, is also inherited as an autosomal dominant predisposition to breast cancer, but is not associated with an increased risk of ovarian cancer. Together, these genes account for a large majority of breast cancer associated with a genetic predisposition, about 10% of all cases of breast cancer. The mutant forms of these genes play no role in sporadic cases of breast cancer, which make up 90% of all cases. Both BRCA1 and BRCA2 encode large proteins that are confined to the nucleus and expressed in many tissues. Expression is at its highest during S phase of the cell cycle. Circumstances, including their common pattern of expression, similar phenotype of the mutant alleles, breast cancer, cellular localization, and experimental evidence from studies in mice, suggests that both genes have similar functions and are involved in DNA repair. Direct evidence for the role of the BRCAI protein in DNA repair also exists. In cells exposed to ionizing radiation, which produces double-stranded DNA breaks, BRCAI is phosphorylated, a sign of molecular activation. Center on identifying the kinase proteins that carry out this phosphorylation, in an attempt to place the BRCA1 and the BRCAL proteins in a pathway associated with DNA repair. It suggests that the BRCAI protein can be phosphorylated by two different kinases and that either kinase is activated by double-stranded DNA breaks, Fig 7. In this model, DNA damage activates the ATM kinase or the CHKL kinase, or both. These kinases phosphorylate the BRCA1 and p53 proteins. The activated p53 protein arrests replication during S phase to allow DNA repair. The active BRCA1 protein participates in DNA repair with the BRCA2 protein, the MRAD51 protein, and other nuclear proteins involved in DNA repair. The role of BRCA1 and BRCA2 proteins in DNA repair may also explain why they are not associated with sporadic cases of breast cancer. In rice, at least 3 mutations would have to accumulate in a breast cell for it to become cancerous. Both copies of the BRCAL or BRCA2 genes would have to be mutant, and at least one other mutation in a cell cycle regulation gene would have to occur. On the other hand, those who inherit a mutant copy of either the BRCAL or BRCA2 gene already have one mutation and need only 2 more to trigger breast cancer. 31.3.2. Oncogenes induce uncontrolled cell division. We know the second category of genes involved in cell cycle regulation, those that normally promote cell division, called protein cogenes. When expressed, these genes promote or maintain cell division. To stop division, these genes or their gene products, or both, must be inactivated. As mutant alleles called oncogenes, they induce or maintain the uncontrolled cell division associated with cancer. The protein products of protein cogenes are found throughout the cell, including the plasma membrane, cytoplasm, and nucleus. In spite of the ranging locations within the cell, suggesting varying functions, all protein cogene proteins characterized to date alter gene expression either directly or indirectly. Unlike repressor genes, where mutations in both alleles of a gene are necessary to promote the development of cancer, only one of the 2 copies of a protein cogene needs to mutate to induce malignancy, resulting in a dominant phenotype. Thus genes capable of causing cancer are known as oncogenes, Greek, onkos tumor or mass. Oncogenes were originally discovered in tumor-causing viruses. These viral oncogenes were found to be closely similar to certain genes present in the normal host cells, which are referred to as protein cogenes. Now, about 40 viral and cellular protein cogenes have been identified. Protein cogenes encode for growth-regulating proteins. The activation of protein cogenes to oncogenes is an important step in the causation of cancer. A. Activation of protein cogenes to oncogenes. There are several mechanisms for converting the protein cogenes to oncogenes. Some of the important ones are described here under. I. Cell insertion into chromosome. When certain retroviruses genetic material RNA infect cells, a complementary DNA, cDNA, is made from their RNA by the enzyme reverse transcriptase. The cDNA so produced gets inserted into the host genome, Fig.8. The integrated double-stranded cDNA is referred to as provirus. This proviral DNA takes over the control of the transcription of cellular chromosomal DNA and transforms the cells. Activation of protein cogene MYC to oncogene by viral insertion ultimately causing carcinogenesis is well known, e.g. avian leukemia. Some DNA viruses also get inserted into the host chromosome and activate the protein cogenes. E. Chromosomal translocation. Some of the tumors exhibit chromosomal abnormalities. This is due to the rearrangement of genetic material, DNA, by chromosomal translocation, i.e. splitting off a small fragment of chromosome which is joined to another chromosome. Chromosomal translocation usually results in overexpression of protein cogenes. Hodgkin's lymphoma, a cancer of human B lymphocytes, is a good example of chromosomal translocation. In this case, a fragment from chromosome 8 is split off and joined to chromosome 14 containing MYC gene, Figure 9. This results in the activation of an active MYC gene leading to the increased synthesis of certain proteins, which make the cell malignant. E. Gene amplification. Several-fold amplifications of certain DNA sequences are observed in some cancers. Administration of anti-cancer drugs methotrexate, an inhibitor of the enzyme dihydrofolate reductase, is associated with gene amplification. The drug becomes inactive due to gene amplification resulting in a several-fold, about 400, increase in the activity of dihydrofolate reductase. IV. Point mutation. The ROS protein cogene is the best example of activation by point mutation, change in a single base in the DNA. The mutated ROS oncogene produces a protein, GTPase, which differs in structure by a single amino acid. This alteration diminishes the activity of GTPase, the key enzyme involved in the control of cell growth. The presence of ROS mutations is detected in several human tumors, 90% of pancreatic, 50% of colon and 30% of lung. However, ROS mutations have not been detected in the breast cancer. B. Classification and mechanism of oncogenes. Oncogenes encode for certain proteins, namely oncoproteins. These proteins are the altered version of their normal counterparts and are involved in the transformation and multiplication of cells. Some of the products of oncogenes are discussed below. I. Growth factors. Several growth factors stimulating the proliferation of normal cells are known. They regulate cell division by transmitting the message across the plasma membrane to the interior of the cell, transmembrane signal transduction. It is believed that growth factors play a key role in carcinogenesis. A selected list of polypeptide growth factors and their sources and major functions are. Epidermal factor EGF, salivary gland. Fibroblasts stimulates the growth of epidermal and epithelial cells. Platelet-derived growth factor PDGF, platelets stimulates growth of mesenchymal cells. Promotes wound healing. Transforming growth factor TGF-alpha, epithelial cells similar to EGF. Transforming growth factor TGF-beta, platelets, kidney, placenta. Inhibitory effect on tumor cells. Tumor necrosis factor TNF-alpha, monocytes. Necrosis of tumor cells. Interleukin-2 helper T lymphocytes stimulates growth and maturation of T cells. The cell proliferation is stimulated by growth factor. In general, the growth factor binds to a protein receptor on the plasma membrane. This binding activates cytoplasmic protein kinases, i.e. phosphorylation of intracellular target protein. The phosphorylated proteins in turn act as intracellular messengers to stimulate cell division, the mechanism of which is not clearly known. Transforming growth factor TGF-alpha is a protein synthesized and required for the growth of epithelial cells. TGF-alpha is produced in high concentration in individuals suffering from psoriasis, a disease characterized by excessive proliferation of epidermal cells. Growth factor receptors. Some oncogenes encoding growth factor receptors have been identified. Overexpression and/or structural alterations in growth factor receptors are associated with carcinogenesis. For instance, the overexpression of gene ERB, encoding EGF receptor is observed in lung cancer. GTP binding proteins. These are a group of signal transducing proteins. Guanosine triphosphate GTP binding proteins are found in about 30% of human cancers. The mutation of ROS protein cogene is the single most dominant cause of many human tumors. The involvement of ROS protein, product of ROS gene, with molecular weight 21 kd, P21, in multiplication is illustrated in figure 10. The inactive ROS is in a bound state with GDP. In the cell stimulated by growth factors, ROS P21 gets activated by exchanging GDP for GTP. This exchange process is catalyzed by guanine nucleotide releasing factor GRF. The active ROS21 stimulates regulators such as cytoplasmic kinases, ultimately causing DNA replication and cell division. In normal cells, the activity of ROS P21 is short-lived. The GACE activity, which is an integral part intrinsic of ROS P21, hydrolyzes GTP to GDP, reverting ROS21 to the original state. There are certain proteins, namely GTPase activating proteins GAP, which accelerate the hydrolysis of GTP of ROS P21. Thus, in normal cells, the activity of ROS P21 is well regulated. Mutations in ROS gene result in the production of altered ROS P21, lacking GTPase activity. This leads to the occurrence of ROS P21 in a permanently activated state, causing uncontrolled multiplication of cells.